All right, we're ready to begin the lab. So make sure that you've looked at my intro video, done the discussion forum, and then looked at the other two 5-minute videos I have. So by this point, you should be well prepared to begin the lab. So we're going to go to the same website that was in the second video that I had. And as we do that, I want you to do the same thing that I'm doing. So I'm going to walk you through this, make sure that you're doing exactly what I'm doing, stop the video as needed. And, at certain points, I'm going to ask you questions. And those questions you should answer those questions, and then you will submit those as your lab report. And I'm going to type these questions up into a Word document so you can download that Word document and enter your answers directly in that Word document if you like. And then what you're going to do is you're going to go to the Dropbox that I'll have available, and you'll copy your answers from your Word document directly into the Dropbox. The Dropbox is going to be set up like a quiz. So when you open it up, it's going to look like a quiz, but there'll be questions in there pertaining to the lab questions, and then you'll just copy and paste from your Word document into that quiz and then submit that. Okay? So if you haven't done so yet, open up that Word document and have that ready. And then I think we're ready to begin. So first thing is to go to this website, and the URL is right up here, gander, Gander, .wustl.edu. So this is the same site that was used in the video that you watched. So we're going to go to the genome browser, and we're going to go to the July 2014 gene assembly and click on go. So this ought to look familiar if you watched the video, the previous video. So let's talk a little bit about what we're seeing here and some of the features that weren't mentioned in the video. First of all, this is what we call a contig, C-O-N-T-I-G. This is a DNA sequencing term, but basically it's a continuous sequence of nucleotides. In this case, it's a continuous sequence from nucleotide 1 through 11,000. Okay, so you'll see the term 11,000 base pairs. That's the size of the contig that we are looking at, and the specific position. We're starting right at the edge of a chromosome and going to the 11,000th nucleotide in that chromosome. All right, so notice the zoom buttons up here. You can zoom in by various factors. You can zoom out by various factors. So you'll want to be able to do that on occasion as we go along. So down here we have the genes themselves. So the video should have explained what these are, exons and introns. We have one gene here, which has 3 different splicing possibilities. We have a gene over here, which has only 1 splicing possibility, or 1 isoform. And then we have a gene over here, which has 2 isoforms. And then we have all kinds of settings down here, and we'll be playing with these as we go along also. Now the next thing that you're going to do is under mapping and sequencing, you're going to set base position to full. And basically what that's going to allow us to do is to see the 3 different reading frames that you can get. So remember we talked about in class, if you read out of frame, then you get garbage basically. So the AUG start codon typically establishes that correct reading frame. But as we're going to see, the reading frame gets tricky here when you go from one exon to the next exon. So pay attention. And if you haven't done so, make sure that you looked at the video on splicing and phases. Phases are going to be very important for what we're doing here in a few minutes. And also we're going to set, under genes and gene prediction tracks, we're going to set fly-based genes to full also. Now, anytime you change a setting down here, you want to hit the refresh button. So I'm just going to click this one down here at the bottom. So now my screen is updated with those new settings. Now, we're going to go up to the search window here, and you're going to type in config 1 9700 through 11,000. Now, from the other video, which is this config that you see right here, basically what we're doing is we're zooming in on the very end over here, 9,700, about right where my mouse is, to the end of this gene. So if you recall from that other video, this is the transformer gene. So we're going to spend all of our time here looking at this transformer gene in some detail. So after you've typed in that config number, just hit enter or click on go. And basically all we did there was we zoomed in on the transformer gene. Now let's remember up here in the left, we have two different isoforms of this TRA transformer gene. There's transformer RB and transformer RA. Okay, so those two isoforms, and as we're going to see later on, one of these is going to be the female isoform and one's going to be the male isoform. So, and remember that the boxes here represent exons. So if you put your mouse on that first box, it's going to say exon 1 of 3. And if you move over to the next exon, that's 2 of 3. And then the last exon, 3 of 3. Similarly with the introns, 1 of 2, etc. Okay? So you can always kind of find your location. Even if you zoom in real tight, you can just mouse on there and you can find out exactly where you're at. Okay, sorry about that delay there. First question is this. And so this is question 1 on the Word document. Based on what you're seeing here, what can you tell about the first intron of transformer RB compared to the first intron in transformer RA? So what can you see that's different? Just tell me basically in just a few words what is different between transformer RB intron 1 and transformer RA intron 1. Pause the video and answer that question. Now we're going to update some settings. So down at the very bottom where it says RNA-seq tracks at the bottom, I want you to click on RNA-seq coverage. We're going to change data view scaling to use vertical viewing range setting. And then max, we're going to change max to 37. All right. And then make sure that adult females is checked and adult males is checked. Once you've completed all that, just go to the top and click on the submit button. And then finally, under exon junctions, we're going to change that to full. Now at this point, we want to make sure all those settings, oh, one other thing, I'm sorry. We also want to set RNA-seq coverage to full. We could have done that on that previous screen also, but it works fine here also. So now we want to make sure that those settings actually take place, so you'll need to click on refresh at this point. All right, so things are going to look very different now. All right. So now notice we've got adult females and adult males. And what these colors are showing us in blue, we have female. This is basically showing us how much RNA is being produced. So in other words, how active the genes are. So the big peaks here means that that area of the gene is being transcribed actively. A small peak, like you see in this region here, means that it's not very active. So notice that this region between roughly 10,000 and 10,150, notice that that region is low for females, whereas it's relatively high for males. Okay, so keep that in mind as we go forward. Also notice that in our two isoforms, transformer B and transformer A, the 5' splice site, which is right there where my mouse is, that's the 5' splice site, because here's the intron. This is the 5' end of the intron. This is the 3' end. So notice that the two 5' ends are the same, but the 3' end for B is here, whereas the 3' end for A is much further down. Okay? So that's the 3' splice site. Those are different, but the 5' splice sites are the same. So now we're going to zoom in a little bit, and we're going to zoom in this region around 9850 to 9860. Now, an easy way to do this is to first of all put your mouse on the number 9850, or just a little bit to the left of it. Click and then drag your mouse a little bit to the right, and then release. Okay, so now we are zoomed in on that region, and I'm just going to click and drag that down. Click and drag it down, roughly somewhere there. You should be seeing about 15 or 20 positions here. So notice these positions here, 9845, 9846. Okay, so that's about what we should be seeing. If you're seeing too much or too little, just use the zoom. You can zoom out, you can see a little bit more, or zoom in to get a little bit more detail. Okay, so get your view looking something like this. Now we notice that we have something new that we can see here. We have 3 rows of shaded area up here, just above the contig. And you will see some letters in there. Any idea what those letters might be? Those are amino acids. So why do we have 3 different rows of amino acids? Well, because there's 3 different reading frames. So take a look, for example, at this CGA between 9856 and 9858. CGA. Well, that codes for arginine. That's what the R is. But maybe that's not the right reading frame. What if the right reading frame is actually GAT between 9857 and 9859? Well, that's going to give you D, which I think is aspartic acid, but don't quote me on that. But of course, that might not be the correct reading frame either. It might be ATG, which codes for methionine. Okay, so how do we determine which one is the correct reading frame? All right, so we have three possibilities. There's always three possibilities because a codon has three nucleotides. So where do we start? Well, as we talked about in class, we start with the start codon. The start codon not only tells us where translation will start, but it also establishes the proper reading frame. So do you see a start codon? So looking at the nucleotides at each position, how would we find a start codon? Well, what are we actually looking at here? These are obviously DNA nucleotides, right? ATs, see the T's in there, A, T, C's, and G's. But are we looking at the template strand or the coding strand? Well, this is actually the coding strand. So take a moment to think about this. The coding strand is the one that doesn't get used for transcription. The template strand is the one that gets used for transcription. We're looking at the coding strand, the one that doesn't get used for transcription. But obviously, if we know the coding strand, we also know the template strand. Now, remember that when transcription happens, we get an mRNA molecule. And how do we determine the sequence of the mRNA molecule? Well, we look at the template strand and we do complementary bases. So if we see a C on the template strand, we put a G on the mRNA, right? And if we see an A on the template strand, we would put a U in the mRNA, okay? So do that, just make a sketch of that, just a few nucleotides, and then compare the template strand, compare the coding strand with the mRNA. What do you notice about the coding strand and the mRNA sequences? So this is question number 2. Question 2 is simply to compare the coding strand of DNA with the mRNA sequence. Are they the same? Are they different? If they're the same or different, tell me exactly what do they have in common and what don't they have in common. Okay, so that's question number 2. So continuing on, to look for a start codon, what we would be looking for is, first of all, we want to make sure that we're in the exon. So again, notice the black box here tells us we're in the exon. And we want to look for the first start codon. So what would the first start codon would be? It would be ATG. Now you may be thinking AUG, well, yep, that's for the RNA. At the template strand of the DNA, it would be ATG. So here's the start of our codon. So we go, though there's an ATT, that A is not in the exon, so TTT, that's not it. We want to look for A. Well, there's an A right there, and that A is followed by a T, followed by a G, and notice that codes for methionine. Okay, so right there, ATG, that sequence, I'm just going to zoom in on that so you can see that better. Oops, I'm going the wrong way. Sorry about that, just a moment. Okay, right there it is, ATG, positions 9858 through 9860. That's our start codon. Okay, and the M there tells us that that codes for methionine. So now I'm going to zoom back out. All right, so there's our start codon. There's our methionine, which is going to end up being the first amino acid in the protein. And we're in the exon. Well, what is everything to the left of this ATG? So that G there, and everything else to the left of that. Let me zoom out a little bit more. So once you get to the left of the start codon, what do we have? We have an untranslated region. Untranslated, not untranscribed. This region does get transcribed. It shows up in the pre-mRNA, but it doesn't get translated because translation starts at the start codon. So that means in order to figure out what the amino acid sequence for this particular gene is going to be, we want to look at the bottom of these three reading frames. So M, methionine, is the first amino acid. The next one, which is coded for by A, that's going to give us the amino acid K, which is lysine, and then so on. Notice we have another ATG, which also codes for methionine. We have another methionine showing up in the third amino acid position. So now let's zoom back out. So we're looking at the entire first exon, which you can see here, and we can now read off the amino acid sequence for the first exon, M, K, M, D. I'm just reading the bottom row here, A, D, S, S, G, T, Q, H, R. Okay? So that's going to be the amino acid sequence for the first exon. So let's go back out. And notice that's the same for both of the isoforms, the transformer B isoform and the transformer A isoform. They both have the same splice site. Now, from our splicing discussion, we said that the intron itself, okay, which is in this thinner line here, the intron has a 5' splice sequence and a 3' splice sequence. All right, so if you need to, zoom in a little bit so you can see what those sequences are. What are the first two nucleotides in the intron? Well, right there they are, G and T. What are the last two nucleotides in the intron, at least the intron for transformer isoform B? A and G. Okay, so GT, AG, that marks the boundaries of the intron. So you may remember the sequences GU and AG from last week's lesson. Well, GU is the same as GT, right? Because once this gets transcribed into RNA, that's going to be GU instead of GT, and then it will be AG. Okay, so those mark the 5' and 3' ends of the intron. Okay, so now I have two more questions for you. Question number 3, give the coordinate. The coordinate is the number at the top, so like 10,000 or 9,950. Give the coordinate for the last base of the first exon for transformer RA. What is the coordinate for the last base of the first exon for transformer RA? And then question 4, give the coordinate for the last base of the first exon for transformer RB. Question number 5. What is the consensus sequence for the 5' splice site? What is the consensus sequence for the 5' splice site? Okay, that's just the first two nucleotides of the intron. Okay, we already pointed those out earlier, the first two nucleotides of intron 1. And then question 6, what are the coordinates? There should be two numbers here. What are the coordinates for the 5' splice site in transformer RA? And then question 7, what are the coordinates for the 5' splice site in transformer RB? Again, there should be two numbers there because there's two nucleotides. Okay, so that's just the coordinates for the consensus sequence that you answered in question 5. Now this next question is the hardest one, and if you've forgotten what phase is, go back and review the video on splicing and phases. So question number 8, what is the phase at this splice site? What is the phase at this splice site? Again, we're talking about that first splice site. So if you scroll in, you'll be able to see this better. Or rather, zoom in, I mean. So if you zoom in at the end of the first exon, the beginning of the first intron, see if you can determine what phase is at this splice site. Now we're going to zoom out again, and we're going to go to the end of the first intron for transformer RB, and we're going to zoom in on that area right there. So I'm just going to, again, just do my click and drag right there at the beginning of the second exon. Okay, so question number 9 is, what are the coordinates for the first base of the second exon in transformer RB? Question 10, what is the consensus sequence for the 3' splice site? Again, that's the last two nucleotides in the intron. Number 11, what are the coordinates for the 3' splice site in intron 1 of transformer RB? Number 12, what phase do we anticipate? What phase do we anticipate? Now remember where we left off with exon 1. And again, 13, this is a tough one. Notice that before our reading frame was the bottom of these 3 reading frames. But is it still the bottom? Or are we in a different reading frame now that we have begun exon 2? So for question 13, I want you to tell me which of these 3 reading frames is the correct reading frame for exon 2. Is it the top one, the middle one, or the bottom one? Now I want to show you one more thing. Notice the asterisk or star, whatever it is that we have here. And I think it's in red. What is that? Well, notice the reading frame here is TGA, right? TGA. Well, what would that be at the RNA level? Well, it's going to be UGA. Well, what is UGA? If you take a look at your codon chart, I think you'll see that UGA is a stop codon. So that's what that is right there, okay? So for question number 14, I want you to, once you've figured out what the proper reading frame is for exon 2, so going back to exon 2, once you've decided what the proper reading frame is, I want you to find the first stop codon. Find the first stop codon. Okay, so this is going to require you to first figure out which is the correct reading frame for exon 2. It may or may not be the same as exon 1. All right, so keep in mind phase. So once you figure out the correct reading frame, I want you to find the first stop codon for that reading frame and provide the coordinates for that reading for that stop codon. And again, that should be 3 numbers. 3 numbers for the stop codon coordinates. Okay, so again, once you've figured out what the correct reading frame is, just scroll to the right, keep moving to the right until you find the first stop codon, and then provide the coordinates for that stop codon. Okay, so now we're going to zoom back out so we can see all of intron 1 for transformer RA. And I'm going to go to the 3' splice site for transformer RA. And I'm going to zoom in on that region right there. So question 15, I want you to answer what are the coordinates for the first base of the second exon in transformer RA? What are the coordinates for the first base? 16, what is the consensus sequence for the 3' splice site? Okay, should be the same as the 2 nucleotides at the end of the intron that we've already talked about. 17, what are the coordinates for that consensus sequence in intron 1 of transformer RA? Number 18, given the phase that we are in, so if you've forgotten that, it says it's back to exon 1, look at the phase. Which of the reading frames are we in now? So question 18 is which of the reading frames is correct? The top, the middle, or the bottom? So again, if you zoom in on exon 2, are we going by the top, middle, or bottom reading frame for exon 2 in transformer RA? Question 19. Once you've determined that reading frame, top, middle, or bottom, look for the first stop codon, if there is one. If there's not, then just say not present. Otherwise, if you do find it, give the coordinates of the first stop codon in exon 2. And now we can finally answer the ultimate question. So first of all, males use one of these isoforms and females use the other one. So again, zooming out here, we see that the 5' splice sites are the same for both isoforms. However, the 3' splice site is different for transformer A. Sorry, this is B, transformer B, compared to transformer A. The 3' splice sites are different. Which one do you think males use? Are males forming transformer RB with the splice site right there? Or are males are they making transformer RA with the splice site down there? So this is where we're applying now what you learned in the introduction video. So in the introduction video, we talked about this whole domino effect of splicing and how that leads to eventually the fruitless protein being produced or not. But remember the transformer protein, this is before we get to fruitless. Does the male have an active form of transformer or an inactive form of transformer? And which of these 2 splice sites, okay, either transformer B or transformer A. Which of those does the male use and which of those does the female use? And just to remind you from that introduction video, this is the image I'm talking about. So remember, this is the transformer. Okay, exon 1, exon 2. There is the female mRNA, and there's the male mRNA. So your job for question number 20 is to tell me, do males which isoform do males form, transformer RB or transformer RA, and which one do females form? Now what we could do at this point is we go on and do the same sort of analysis for exon 3, and we could even figure out what the complete amino acid sequence would be for both transformer RB and transformer RA isoforms. But we won't go through that exercise at this point, but of course you're welcome to do that if you like. So we will conclude this lab at this point. So again, type up your responses to these 20 questions in a Word document and then copy and paste them into the Cobra Dropbox. That's it for now. Bye-bye.